Okay, guys, so we just continue on with, sorry, then I just continue on with the chapter 18 of the bio, and then we'll finish with this chapter. Okay, hold on. Okay, so the next thing that we learn, it will be test crosses. Test cross means, okay, when you see an organism, let's say, for example, a black fur rabbit, okay, and then we say that this black fur rabbit is dominant, but we don't have a microscope to look at each of the cells to see whether it's a homozygous, heterozygous, right? We cannot do that very easily. So when you see a black fur rabbit, it's either homozygous, dominant, or heterozygous. It can be, it's black color, but it has also a white allele inside the genes. We don't know, right? So that's why we will do this method called test crossing. Okay, we can find out the genotype of an individual with the dominant phenotype by crossing it with the homozygous recessive genotype for this gene. Okay, so this is called test cross. Now, what does this mean? It means, for example, we want to find out if let's say, for example, a tall pea plant. Okay, we want to see whether it's a homozygous or heterozygous pea plant. Is it pure 100% have tall alleles, or is it it's tall, but it also has short allele inside the genes? Okay, so what we do is we will let it mate together with the homozygous short pea plant to find out. Okay, so let's assume okay, let's say the tall pea plant is really 100% tall, so it's homozygous. So if you cross it, all the children will have the dominant gene, means dominant phenotype. It will show dominant. There's totally no short pea plant. For example, you see this one, big T, small t, right? This is homozygous recessive, right? So once you cross it, even though there's heterozygous children, okay, but all of the children are 100% tall, okay? But let's say this pea plant is not homozygous, is heterozygous. Once you pair it with a homozygous short, and then definitely it will come out, the offspring will have the shortness there because he himself, the tall pea plant itself, has the short allele. So once you see the offspring, there is this short feature, we can say that, oh, okay, actually this pea plant is not pure, it's not 100% tall, okay, it's heterozygous. Okay, so it's very useful to use test crossing, but of course it takes time. You have to wait for the animals to, plant or animals to mate, then see if it's successful, and then you have to make quite big numbers, okay, because probability, you want to see probability, okay, you want to see how much of the kids really have this tendency to show recessive, okay? Again, so after the test cross, if the offspring shows a recessive allele feature, it means that the parent organism you're experimenting on is heterozygous. So this is important because when essay time, they ask you, how to determine whether this father is a homozygous. Then you have to write down by test cross. Then you have to explain how, or you have to test cross it with some information. Just based on these notes lah. Okay, later you see the question. The last one that we're going through is the sex inheritance. So many of you know that we have sex chromosomes, okay? For male, you have XY chromosome, female is X and X, okay? So females don't have Y gamete in the womb. It's only carried by the males. So sex inheritance largely depends on which sperm reaches the egg, okay? Because only the father has the Y chromosome. So if the sperm that has Y chromosome fertilizes the X chromosome, then the baby is XY, then it's a boy. So boy or girl really depends on the father, not the mother, because females only produce X gametes. Okay? Right, so let's say you're pregnant. You want to know whether there's a higher probability of having boy or girl. So there is no such thing as high probability of having boy or girl. Everything is 50-50. Okay? So you can determine that by doing the test cross. Okay, so this is the father's chromosome, X, Y, mother is X, X. Once you do the test cross, you see that it is quite fair, 50% chance female, 50% chance male. Right, next. So sex linkage, this is regarding the disease. Some diseases are more common in men than women. Now there's a reason for that, right? So you look through the chromosome. For female, XX, the X chromosome is longer, but the Y chromosome is shorter. Okay, what does this mean? So it means that genes on the X chromosome, sometimes you cannot find it here. Like for example, half of this is not in the half of that Y. Okay, and some genes found in the Y cannot be found in the X. Okay, so it means a woman never has a copy of these genes, and a man has only one copy. Just to let you know why lah. Okay, because the size is different. Okay, now let's go through the one of the disease. Okay, red-green color blindness. When you're doing sex-linked disease, remember to always use your XY chromosome. So for female, it's X and X. Then beside the X or Y, you write down the, the name, the allele, okay, for the sickness. So let's say if you're if it's normal without color blindness disease, we use the capital letter B. We just use whatever random letter lah. So for me, I'll use capital letter B. Okay, small letter b means there is a red-green color blindness allele there. Okay, let's go back. So woman is X, right? So X big B means this woman has normal vision because X big B is dominant, right? Okay, next one. X big B, X small b. Small b is for red-green blindness. Now, you see that even though she has the red-green blindness here, but because the dominant allele is the big B, meaning the red-green blindness won't show in this woman. Means she's not affected, she's a normal vision person, but she's a carrier. Carrier means that she has this probability that she will give to her kids this red-green color blindness. Okay, but she is with normal vision. She doesn't have the disease. Okay, we call this person a carrier. Sometimes carriers don't know they even have this allele. Okay, now if the woman so happens have two of this small b, okay, there is no dominant big B, so it will show of the feature of the red-green blindness. So she kena already lah. Okay, now for men there's X and Y, right? So if the X chromosome has this big B, okay, he has a normal vision. If he has this X small b from his mother, kena lah. Okay, man with red-green blindness. There is no other X chromosome to help him back up or to help him shield the disease. Now, once he has this small b, then definitely he kena red-green blindness. Okay, so it's more common in men than in women. Because even if the woman has this small XB, might be a normal carrier, normal vision and carrier, okay, or kena the red-green blindness. So let's do this test cross. So what happens when a woman with normal vision, but unknowingly is a red-green color blindness carrier, marries a man with normal vision? What is the probability of their child getting the disease? So write down your chromosomes. So parent one, the mother, okay, XB, big B, small b. Then the father, X big B, is normal, okay, normal vision, Y. So once you do the crossing, you'll see that the daughters, let's look at daughters. One of the daughter would be normal, one of the daughter would be carrier. This one, okay, if they have a son, 50%, he'll be normal, no effect, or he will have red-green blindness. So that's quite a big risk to take lah. Okay, so that's it for sex linkage disease. I will give you some homework. Okay, and then that's all for this chapter. We can go to chapter 19. Right, thank you.